Good evening or afternoon, whenever you're watching this. This is the lecture on forensic entomology for JS 110 Introduction to Forensic Sciences, taught at the University of Alabama at Birmingham. Future 2017 Bahama Bowl champions, am I right? Which brings up a good point. This lecture is being recorded in the fall of 2017. So if you're accessing this archive lecture in future semesters, all the material about forensic entomology still applies. But if I mention anything about the Bahamas Bowl or anything else pertinent to November of 2017, you can ignore all that information. Or if I mention anything about really quizzes or exams and stuff like that. No matter what semester you're taking this, I imagine I'm going to keep the format somewhat similar, where we're hitting this forensic entomology late in the semester, where we've already talked about the traditional types of evidence analysis, biology, chemistry, done in a forensic science laboratory. And now we're moving on to these related fields to forensic science, where you may still get an expert witness, in this case an entomologist, coming to court. But as we learned in one of the first couple lectures, the education for a forensic entomologist is vastly different. You know, with my forensic DNA and forensic science background, I couldn't go into court and testify as a forensic entomologist, even though I did a little bit of research in the area of forensic entomology, getting DNA out of megagut contents. And furthermore, I couldn't apply to a entomology agency next season on Fox. Bugs. A semi-attractive forensic entomologist teams up with and then you can fill in the rest on your own. Because the education for a forensic entomologist is where they get their undergraduate degree in biology, and then they go to graduate school for specifically entomology, and they spend their entire research career sort of focusing on entomology and learning about the growth rates of maggots and pupa and flies and beetles, and that's basically what we're going to talk about tonight. All right, so I asked some people to throw, Sarah, always reliable, threw a quick, she knows how this works now, right? She threw a sentence: a dead body's found at home and there's flies present on and around the body, and a forensic entomologist is called in to determine time of death, okay? And Sarah's really hit on all the major points. Really, when the forensic entomologists come in, it's when a dead body is found. And normally, with time of death, the pathologist is asked to give that estimation. But if there are a lot of maggots or flies or other immature insect samples on the body, what may happen is that these maggots and flies, here, let me draw a little fly here as well. Oh, I can't draw a fly. Will be collected and sent to an entomologist, and they will also give an opinion on a time of death that, when using insect evidence, can oftentimes be much more specific than the forensic pathologist. And I may have mentioned, depending on the version in forensic pathology, that we have Greg Davis, the speaker, come in and speak to our program often, and he hates estimating time of death as a pathologist just because there's so much variation in the way that a body can decompose. There's a little less variation, or it certainly helps if the pathologist and the forensic entomologist come to the same conclusion about time of death. So really, bullet number 2 there, estimating the time of death, that's a large part of what a forensic entomologist does. And they do this by estimating the postmortem interval. Really, these last two things are very similar. And that's what most of this lecture is going to be about. I mean, we're going to have some introduction to the types of insects and flies and beetles that are found on dead bodies. And then that first bullet, occasionally a forensic entomologist will be asked to do other things besides estimating the time of death. So we'll talk a little bit about some other things that a forensic entomologist may be a part of. All right. The definition of forensic entomology, so in case you haven't figured out, entomology is the study of insects and their arthropod counterparts, okay? Bugs, if you want to think of it that way. And forensic entomology is studying these bugs, how they interact with legal matters. The first thing to remember is that a forensic entomologist, although most forensic entomologists are concerned with this last one, the time of death estimation, depending on their specialty, some forensic entomologists will be called in to help. Where did I lose my cursor there? Help with some things that don't involve dead bodies. For example, as we can see here, termites, bugs in home, bugs in food. If you find maggots in your hamburger when you go to Wendy's or Arby's or I don't care whatever fast food restaurant you think is so awful that they may have maggots in their food, a forensic entomologist may be called in to testify about those maggots, whether it's possible that those got into the food during the process or during the processing of the food, or maybe it's just the defendant or the person who's suing the restaurant. They sort of threw them in there on their own. I mean, we had a similar case when I was doing my dissertation research. My advisor was a forensic entomologist, and I don't know if he ever took the case, but he was involved in a case where it was bugs in food, and we were discussing it, and he said where if the maggots on the food were the type of maggots that are traditionally found on meat, then it's very unlikely that the eggs and the maggots could have made it through the process of the processing of the beef, the freezing of the meat, the cooking of the meat. It's not as though if the eggs were laid at the meat plant that you would cook a hamburger and all of a sudden they would hatch and start feeding and be gigantic maggots by the time it got served to your customer. But if they were the type of maggots that fed on lettuce or vegetables, well, it's a lot more likely that they may make it through the food manufacturing process. So an entomologist, like you and I, we look at a maggot and I don't know, a maggot looks like a maggot, but forensic entomologists can actually determine the species and the habits and where these insects are commonly found. All right, a move body, wounds, or drug use, it's just worth mentioning. If you move a body from like urban area out into the woods, you may find a species of fly that has developed on that body and has hatched from the pupa that is an urban fly, one that's only found in the city. Well, that may help the investigators determine that the body was killed in one location and then moved to another. If you find a maggot mass around sort of in the chest cavity, as we're going to find here, most maggots grow in the moist parts of the body. So if you find a big mass of maggots like directly in the chest, that may indicate that there was a gunshot wound or something present. And then also drug use. Sometimes you can, I mean, this is more CSI fantasy stuff, but you may be able to find a maggot and determine if a drug is in the body that the maggot was feeding on, then it will be in the tissue of the maggot also. So it has to be a weird situation where you find the body and all the soft tissue has been consumed. The blood is not viable for testing, but maybe you can determine that a drug was present by sampling the maggots. All right. Time of death. Okay, that's the big one. And what forensic scientists, forensic entomologists are trying to do, they're trying to determine the postmortem interval. I'm going to draw this little picture here several times, but the postmortem interval is the time between death and discovery. Okay, and that's where we have this dead body. I'm not doing a very good job drawing it again. But when the body is discovered, then there's a bunch of maggots, or maybe there's flies around it, maybe there's pupa, but there's a bunch of bugs growing on the body. Okay, and we'll draw the little police officer here with the hat. I don't know why a police officer would wear that hat, and I don't know why he's smiling either. But there he is. He discovered it. Oh no! There, that's okay. Well, congratulations on the nightmares you'll have tonight. Okay. So they're going to use the insects and other arthropods to answer the question of when did this death occur? Okay, the how or where death occurred, we already talked about that a little bit, with the moving of a body or the gunshot wounds. All right, this lecture is one of the longer ones. And when we do it on campus, depending on how quickly we've moved throughout the semester, sometimes I'll take a little bit more time on this slide. And basically do really poor accents of a person in France. Wait, where's the? Oh no, yeah, Magnan is he's from France there. But you don't want to hear my lousy French accent, so I'll just summarize these very quickly. And China is one of the very earliest instances where it's recorded in a book, the Washing Away of Wrongs. That talks about a lot of other criminal justice related material, but it talks about this one case where there was a murder in this farming village, and they had all the farmers come and lay their sickles down, and the flies started buzzing around one of them, indicating that it had traces of blood and tissue on it. And then, like any good, even back in the 13th century, the story ends with a confession, and they rolled the credits. Okay, so some things never change. All right, and then in 1890, Magnan was the first one to document how he looked at the pupa and the larva and the empty pupae casings on a baby that was found in a chimney to determine that the baby was there for quite a while, and it was not the fault of the individuals who had recently moved into the house and discovered the baby. But that was the first instance, of using insects to determine time of death, or to help estimate time of death. And then a long time passed, and eventually the field of forensic entomology realized that if you're going to look at flies on a dead body, or maggots on a dead body, or pupa on a dead body to estimate the time of death, you need to know for different species of flies, how fast do these maggots grow? You know, if I find a 15 millimeter maggot on a dead body, does that mean it's 3 days old, 5 days old, 7 days old? Okay, what determines it? Does the temperature matter? Does the species matter? Does the rainfall? Does the light day-night cycle matter? So Hall was the first individual to take some blowflies, which, as we're going to see, are often found on dead bodies, and basically raised them in a cage in labs under controlled conditions, and recorded growth data as far as how fast these flies grew. Okay, so this continued with other forensic entomologists collecting this data. Not a lot was done, but then there was sort of a boom with forensic entomology in the 70s and 80s with Greenberg, Marchenko, and they had a lot of students underneath them who started collecting and publishing all this forensic entomology data, to where we are right now, where there's a lot of growth data available to forensic entomologists, where if they happen to find a sarcophagid bellata fly on a body that they're consulting for a case, they can go to the scientific literature and look up the growth rates of the sarcophagid bellata. All right. In 1996, the American Board of Forensic Entomology was formed. This is very similar to some of the other boards that we talk about at this portion of the course, where it's a group of forensic entomologists, and they will look at an individual who sort of applies to be a part of the group, the American Board of Forensic Entomology. They'll look at the education, they'll look at the work experience, and they'll sort of certify these forensic entomologists as a way of saying, yes, this, according to us, the official Scientific Society of Forensic Entomologists, this individual is qualified to go into court and testify. It helps preventing some, backyard entomologist or somebody who got an entomology degree and then just sort of started taking casework from going into court and giving an opinion that they're not qualified to give. So we say insects and their arthropod counterpart. What is this? Okay, what's an arthropod and an insect? Well, back in, elementary school or something, at some point you probably had to say kingdom, phylum, class, order, family, genus, species. And it seems to me from talking to students that everybody sort of had a rhyme that went along. Mine was King Philip came over from glorious Spain. But like everybody's rhyme is different. I can't figure out why in the education system in the United States, all teachers choose a rhyme to help their students memorize the order of kingdom, phylum, et cetera, et cetera. And now it starts with domain. So I don't know, what is it, dumb King Philip or something like that? But then nobody can agree on the rhyme. Could we please have standardized rhymes in this country? Anyhow. Is that not in the Common Core? I don't know much about the Common Core other than people hate it or don't mind it. Get a decent rhyme in there. Okay, so arthropod is the phylum. So when we talk about entomologists deal with insects and their arthropod counterparts, entomologists, if they find spiders associated with a dead body, centipedes, okay, they can give an opinion on why they're present at but the thing is, with all of these other non-insects like spiders, scorpions, centipedes, they may interact with a body if you drop it right on the spider nest. I don't know, do spiders have nests? Okay. Kind people can often follow ghostly screams. I like that. That's a lot nicer than making fun of poor King Philip. All right. So Sarah, did somebody in the class make up that? I'm sorry, that's always my follow-up question. I wonder if like the classes have like contests where the kids are making them up, because oftentimes it ends with spaghetti, which I think is just ridiculous. Okay. So spiders, scorpions, they're not going to seek out a dead body in order to feed or mate on the dead body. But like I said, if you drop a dead body on a spider's nest, then the spiders may wind up doing something, spinning webs on the body or something like that. The same thing when it comes to insects. Not all insects. Ants are not going to search for a dead body and specifically build their ant hill on top of it. But if you throw a body on a hill of fire ants, okay, you can bet that they're going to interact with that body, be found on the body when it's discovered. Okay. Now, of all of these groups, there's really, two of them, and I didn't do my bullets very well here. There's two of them that are more important than the others, and those two are, I don't know, are they still in? They're there. Hey, look, it magically corrected itself. Those two types of insects are flies and beetles. You don't need to remember Diptera or Caloptera. Okay, you can just remember it as flies and beetles, because flies and beetles will search for a body, because a body is a very good place for them to mate, feed, and lay their eggs, because they're young, as we're going to find out. Flies and beetles go through complete metamorphosis, lay eggs, and which hatch into larvae, are specifically designed to feed on decomposing flesh. Okay, how's that for hardcore? All right. Now, even within flies and beetles, not all flies and beetles are attracted to dead bodies. If you find a fly in your kitchen, don't go, oh my God, somebody has hid a dead body in the crawl space, and call the police. It's probably just there for garbage. Okay, if you find one in your backyard, nobody has snuck into your backyard and buried a bunch of bodies. It's probably just poop, animal poop. Well, I don't know. Hey, what you do in your backyard is your own business. Okay. But flies are attracted to garbage and feces and dead bodies, decomposing human remains. Okay, so the flies that are attracted to dead bodies are called carrion flies. And even within the groups of muscid, house flies, fords, not all of these are attracted to dead bodies. There's a lot of flesh flies that are attracted to feces. But within this group, there are two that are a little bit more common than the other ones, and those are blowflies and flesh flies. A large portion of blowflies are attracted to dead bodies, a large portion of flesh flies. If you were to gamble, if you were to kill a hobo, no, you don't go killing hobos. But if you just happened upon a dead body and you wanted to do something and you threw it out in a field in the summer, okay, and you were gambling, I don't know who you're gambling with, on what type of insect is going to appear at the body first, blowflies and flesh flies would be a very good bet, because they're going to show up. Okay. One thing I do want to mention about the phorid flies down here, okay, because I think there's a multiple choice question about phorid flies, and I guess we'll get to it after we talk about the blowflies and the flesh flies. But the phorid flies aren't used that much in forensic entomology, but in a certain circumstance, they may be useful information. May provide useful information. All right, the blowflies are these shiny flies, right? You've probably seen them at picnics, and again, it doesn't mean that there's a dead body in the garbage can at the, Crestwood Park or whatever. But they're called, their nicknames, I don't know if you call them a nickname, blue bottle and green bottles. So that's, if you're a cool entomologist, that's how you refer to them. But it's because they're shiny, they look a little bit like glass, right? They're colorful, they can be a bronzy color. These tend to be the blowflies that are among the first to arrive at a corpse. They're just a little bit more abundant than the flesh flies, which we're going to see in the next slide. All right, here in Alabama, if it's the summer, the green flies and a couple bronze-colored flies are much more active. But then as it gets into the cooler months, a lot of these flies go into diapause, which is sort of like a hibernation state. And you'll find more and more blue flies, like Califorus, you guys don't care about species name, but you'll find these blue bottle flies around dead bodies, and I have in the past and even currently for camps and some classes gone out and attempted to catch flies, and you can just use like a rotting hamburger meat and it will attract these same types of flies. So the flesh flies are large gray flies. They're not as big as horse flies, but they're still, if you saw it, you would say, wow, that's a big fly. Okay. So they have this a white and black stripes on their back here, but some other flies have that too. The really distinguishing feature for sarcophagid flies is, again, most of them are a little bit bigger than a normal house fly, and they have this sort of checkered pattern on their back. Okay, so this is another type of fly that may be found, and there's several species of blowfly, several species of flesh fly. All right, the forward fly. So this is what I mentioned. There may be like one multiple choice question on the quiz or exam about a forward fly. These forward flies are small, and they're called scuttle flies or coffin flies. You may have seen these at picnics. They sort of land, and it looks like they're jumping around on the table. They fly and then land again, so they sort of have this almost hopping-like appearance. It's almost like they're scuttling around the table. That's why they're called scuttle flies. Coffin flies, and this relates to where these flies may be useful to a forensic entomologist. Let's time travel a little bit. So we're going to go back to the Old West, when men were men and so were women. And if you died, they didn't bury you in a fancy coffin. They put you in a pine box, right, with like a bunch of like gaps in it. Okay. And they buried you 6 feet under, or if it was a hot day, 3 feet under. And what they would find is, inevitably, they would bury somebody. Okay, now it's going a little bit off the rail. They would bury somebody, and then like the coffin maker or the grave digger or somebody would go, oh my goodness, I left my digital wristwatch inside the coffin. So they would have to dig up the body. Okay, so they dig up the body. And occasionally when they dug up these coffins and opened them up, what would happen is either a bunch of flies would come out, okay, or they would find a bunch of dead flies in the coffin. So the question is, how in the heck did all of these flies get in the coffin? They weren't there when the body died, okay? And it's much too many where not all of them independently like started burrowing through the ground to get to the body. What happens with these coffin flies is one female gets to the body and lays eggs, and then those eggs hatch, larvae, they go into pupa, they hatch into flies, and if the body's still buried, then the flies don't have anywhere to go, and they just die in the coffin. Okay. How does this, wait, we don't use pine box. Whoa, where are you going with this, Dr. Linville? Where I'm going is these forward flies are able, if it's a shallow grave, to, get through the dirt, and it prevents a barrier for other flies like the blowflies or the flesh flies from getting the body. So you can imagine if there's some type of physical barrier, if there's a body in a trunk of a car and the trunk is sealed pretty tightly, or if a body's wrapped in plastic, next to the river, then it may be a situation where the larger flies, the blowflies or flesh flies, don't get to the body as quickly as these forward flies. So therefore, if you're a forensic entomologist and want to estimate the time of death, it becomes advantageous to look at the forward flies on the body, or maggots or pupa on the body, as opposed to the blowflies or flesh flies. Sometimes it might be the only fly that's there. Okay, so it's important that forensic entomologists do study and know a little bit about the forward flies. Although, if there's forward flies and blowflies on a body, you're going to want to work with the blowflies because the maggots are a lot bigger. Did I mention these forward flies are extremely small? And it's a lot easy to measure and determine the difference between like a 15 or 14 millimeter maggot versus a, 1.5 versus 1.55 millimeter maggot. Oh, not those beetles. Okay. All right, so we're going to move on to beetles. That's just a little background about flies. Okay, with beetles, there's a wide variety of species, so we're not even going to break this down into like the 2 major families of beetles that are found associated with the dead body. And different species will arrive either early or late. Some species of beetles will like to arrive early in the decomposition process. Some will arrive late in the decomposition process. For the most part, beetles are associated more with the later part of the decomposition process, because the flies, if you throw a dead body in a field and have to bet whether a beetle or a fly is going to find it first, the flies can cover a lot more area. So they're they're not designed they're intelligently designed. No, that's not. But the flies will lay eggs, and their larvae consume fresh, soft tissue, much better than any other type of tissue. Well, the beetles, by the time they find the body, all that soft tissue may be gone. So a lot of beetles, their larvae are designed and are able to consume sort of the leftover, the harder cartilage or ligaments or tendon part of the body. All right. So a lot of the beetles will sort of come in and clean up it's not like in the Mummy where you throw a bunch of beetles on a body and then the adult beetles eat all of the flesh. No, if you want to clean up a body with beetles, lay some beetle eggs on it and let the larvae do the work. Although I guess is Brendan Fraser back yet? Is that happening? Has that already happened? I don't know. That's not mentioned this here. We're going to look pretty stupid when somebody's accessing this archive lecture in 2018 and they realize that Brendan Fraser's just won his third Oscar, and we're sitting here wondering if he's going to make a comeback. Am I right? Where were we at? Talking about the Mummy. The Mummy Returns, and now Complete Metamorphosis. Not the movie, but the process. Flies and beetles undergo we learned about this with butterflies, right? The beautiful butterfly lays an egg, and then out pops a beautiful caterpillar, and then it goes into a chrysalis, all right, and then it hatches into the beautiful. We have pretty names for the larva and pupa stage for butterflies, but for flies, it's just eggs, larva, pupa, and adult fly. And sometimes the larvae are called maggots, which that's really not beautiful. That's sort of disgusting. But they do an important job of, consuming the flesh of dead organisms. So there's just not a bunch of rotten bacteria, diseased carcasses around. So they do serve their purpose. But it's important to understand that when a body dies, the egg or the pregnant female flies will be attracted to the body. They lay eggs on the body. And just like in the opening credits of True Blood, if anybody of you are unfortunate enough to watch that show, the maggots hatch and they quickly feed on the soft tissue. So if investigators find a dead body that's been dead for, 3 days or 7 days, there's a good chance that body is going to be covered in maggots. All right. And oftentimes, especially if the body is killed somewhere, after 3 to 7 days, the body starts to stink. And then it's more likely that somebody's going to notice that there's a dead body in the area, and investigators will come up upon it during this time period. But when it comes to the investigation, it may be very important to know whether that body was dead 3 days versus 7 days, versus 12 days. And that's where the forensic entomologist can come in, look at the types of eggs, maggots, larvae on the dead body, and determine how long it's been there. So now we want to look at how these entomologists estimate the postmortem interval. Okay, they collect insects, both adults and immature, but hopefully at this point you're realizing that it's really these immature insects, the eggs, the maggots, and the pupa, that are more useful to the investigators for estimating the PMI than the adults are. The adults just tell them who's in the general area, but really what you want to know is, hey, if I find a 5-day-old maggot on the body, that means the body's been dead for at least 5 days. Right? So that's where they can be very useful. Okay. How are these insect samples collected? If you're collecting maggots and eggs and pupa, they can be collected at the crime scene. It really depends on whether the crime scene unit is equipped and trained on how to collect insect samples. Most of the time, even if you find a bunch of maggots on a dead body, it may not be important to the investigation. Okay, maybe the person wasn't murdered, they died naturally, or maybe there are other factors where when the case goes to trial, the time of death is really not an issue. Like everybody knows that the person died and went missing on a certain day, so you don't really need a forensic entomologist to come in and testify on that. So what often happens is the flies will be zipped up, or excuse me, the body will be zipped up. And then when it gets to the medical examiner's office, if the medical examiner office unzips the body and sees these insect samples on the body, then they are trained and equipped on how to collect these samples. All right, so they'll pluck a couple of the maggots out. All right, now what we see here is this picture. Where did my cursor go? But there we go. What we see right here these are a bunch of maggots that are feeding on human tissue right here. All right, but this is not a dead body. All right, and this goes to something that we're going to talk about here in a little bit. Remember when we talked about the postmortem interval that we're estimating, and it's from time of death to discovery? All right, there's another very important event, and can anybody guess and throw this event into the chat box? What happens in between death and discovery that a forensic entomology would really care about? Okay, in other words, when we find the body and there's a bunch of maggots here on it, okay, what else has to happen? Can anybody take a guess and answer it in the chat box? The body's dead, okay, and then later we find the body with a bunch of maggots on it, maybe pupa. In order for there to be maggots or pupa or eggs on the body, what has to happen? I mean, a body doesn't spontaneously generate maggots, right? I think somebody put a bunch of rotting meat in a jar and proved that a long time ago. All right, the answer is somewhere in here. We have the dead body and the fly has to lay eggs. Okay, now granted, this looks like a bow tie bombing the body, but you get the general idea. Okay, the fly has to lay eggs on the body. All right, now why are we bringing this up right now? Because it's important to ask the question, okay, if flies have to lay on the bed, is it ever possible that the flies would lay eggs on a body here before the person is dead? All right, and the answer to that is no. Well, most of the time, no. Okay, later we're going to come back to this and I'll tell you one instance of why that's true. But the reason is that for most species of flies, the maggots will not feed on living tissue. They will only feed on dead, decomposing tissue. Okay, which brings us to here. What the heck's going on with this? If you want to look up something called maggot therapy, what's going on here is this person has an infected, which looks like it's partially amputated limb. And they had an infection in the end, and oftentimes with amputees or with diabetics or with people who are bedridden, can't move a lot, get bed sores, they have very poor circulation to their extremities. And normally how you fight an infection is with antibiotics, okay, oral antibiotics or an IV. The antibiotic gets into the bloodstream and the blood takes the antibiotic to the infection and the infection clears up. Well, what if there's poor circulation and not a lot of blood going to an area of the body? It's really hard to treat that with oral antibiotics or to treat it with even an IV through the blood. So sometimes when these sores get so infected and the antibiotics, the oral antibiotics aren't working, what they can do is they can actually put very small maggots, the type that will feed on dead bodies, into the wound. And those maggots will only feed on the dead, decomposing flesh, and they will not start eating the living portion of the person. So what happens is you put some maggots on there, you cover it up with gauze, and then the doctor comes back in a day, opens it up, checks it, maybe cleans it up, puts clean, and gradually these maggots do clean large wounds, and they're very effective at doing so. The only downside is that you have maggots living on your body, which probably isn't, the greatest feeling in the world. And all things created equal, if you had to choose between taking an antibiotic pill or having maggots pressed into your flesh, you'd probably choose the pill. But it's sort of worth mentioning that maggots can be used for this maggot therapy, and the reason it works is because these maggots only feed on decomposing flesh. All right, so that's interesting story number one. Okay, you're the medical examiner. How are you going to collect these maggots? Okay, ideally, you're going to kill half of the maggots, preserve half the maggots. Some medical examiners will just preserve all of them. Okay, but let's look at the benefits here. Okay, the reason why you want to preserve them, stop them from growing, kill them, whatever term you want to use, is because you want the appearance of those maggots when they're sent to the the forensic entomologist, which might not be for another couple months once the investigators determine that time of death is an issue and they want a professional to evaluate it. You want to preserve them, stop them, so they appear in 2 months the same way they appear on the day you pulled them off the body. Okay, so by putting them in ethanol or a solution called kale solution that has a little bit of formaldehyde in them, that kills them, stops them from growing, and prevents them from degrading. Okay, this should be ethanol, not ethanol. Somehow these two got reversed, and I haven't been able to fix it yet. All right, so putting them in these solutions, if you just pulled the maggots off and squish them, which some students suggest that's how you should kill maggots. No, don't squish them. Or even if you froze them, okay, and just put them on the shelf, what would happen is they would eventually degrade. All right, so you need to preserve them in some type of preservation fluid. Okay, also, the entomologist is going to want the climatological data, basically the temperature information from the crime scene. Now, the good thing is that this collection, it doesn't have to be done right at the crime scene. You don't need to take a thermometer and stick it in the body and stick it in the ground and, stick your finger up in the air and determine what the good thing is that there are enough weather stations spaced throughout the United States, and this information is publicly available, that most of the time the forensic entomologist can just access this information from the weather stations, even if it is, 6 months after the fact, and they can triangulate and figure out what the temperature was where the body was found. Okay, now, in theory, what is suggested by forensic entomologists, to make it easy on everyone, is to keep half of the maggots recovered from the body alive. Why on earth would you want to keep them alive? The reason is, one of the first steps a forensic entomologist is going to do is they're going to try to identify the species of the fly. But the problem is, if you look at the maggots, a lot of different species of flies look identical when it comes to looking at the maggot. Okay, you can't really just look at a maggot and find morphological features and determine that it's a Chrysomya megacephala versus a Phormia regina. However, if you allow those maggots to grow to the adult stage, then the morphology, it makes it to where you can very easily determine the species, and the species allows you to go into the scientific literature and look up the growth data. Okay? So we don't want to keep all of these as maggots. Okay? We want to grow them up and raise them to adults. Okay, now what if the medical examiner is lazy and just preserves all the maggots? Well, there is a way to identify the species even without the adult, but having the adult is certainly the easiest way. Okay, once the evidence is received, whether it's flies collected from the body, beetles, or flies, beetles over here, or maggots right here, or pupa, the first step is that the entomologist needs to identify the species. Okay, and how is the species identification? Well, okay, wait a minute. Why do we need to identify the species? Well, hopefully this is a little bit obvious here, but different species grow at different rates. So in other words, if we recover a 7 millimeter maggot from decomposing human remains, okay, that maggot may be anywhere from 3 days old to 10 days old, okay? In other words, Chrysomya megacephala may reach 7 millimeters long after 3 days, whereas Phormia regina may take 10 days to reach the length of 7 millimeters. So you need to know the species so you can go to the proper growth data, because the growth data for different species is going to be completely different. Okay, so that's why we need to identify the species. I mean, this sort of shows two closely related flesh flies. one may take 50 hours to hatch into a larva. The other one may take 40 hours. Okay, to hit the pupa, it may take 110 versus 90. This is just shown that these times can be a little bit different. Okay, now how do you identify the species? Well, like I said, if it's an adult fly, most forensic entomologists are trained on how to look at the morphology, the color of the fly, the hairs on the fly, the bristles on the fly, the wing structure, okay, to determine what species the fly is. Okay, you ever see Beetlejuice where there's that, he offers the fly a Zagnut bar and there's like that close up and the flies look disgusting, like you can see all the, or if you've seen the movie The Fly, although that's not quite as accurate because it's a Jeff Goldblum size fly, but flies under microscopes, they look disgusting. Okay, they have all these interesting features. And you can look at those interesting features and determine what the species is most of the time. Now, this male genitalia, no, this is not a joke here. Yeah, flies do have genitalia. There are male flies and female flies. And many of them mate through what's called a lock and key mechanism. And this is like going to be sort of like a very poor drawing of, but it's sort of like the male abdomen, and then it's sort of like a little hook that comes out, which is what the penis organ looks like in males. So the shape of that organ determines whether it will fit into the female, and then fertilization can occur. Because what will happen is two closely related flies might not even be able to tell each other apart. Okay, so if I'm like a green blowfly sitting there and I, at a body hanging out to mate, I guess that's where I go, and I see a female flying by who's green, I may go, well, it looks like a green fly. Let's go try to mate. So I go and we try to mate, and if our parts don't fit together because we're the wrong species, okay, then I'm like, oh, sorry about that. And she's like, it happens all the time. And then we go our separate ways. All right. So it prevents, Formia reginas from mating with Chrysomya megacephala, and it keeps the species separate. Where if I go and try to mate with a green fly and we are the same species, then the genitals fit, and there you go. 9 months later, it doesn't take 9 months for a fly, I don't know how long, but later, okay, fertilized eggs, little baby larvae running around. Okay, so because of that lock and key mechanism, the shape of the male genitalia in closely related species is shaped differently. Okay, and actually when we were working with flesh flies back with my advisor, we were doing some DNA analysis, DNA identification of the flesh flies, and we would send the abdomens to, I think, Thomas Pape, P-A-P-E, who was in some Scandinavian countries, and he was like the expert on looking at flesh fly genitalia. Okay, I don't know if he's retired or not, so if you guys or if anyone's looking for, you want something to be the best that you can be in, the job of looking at flesh fly male genitalia, that might be open for you right now. You only have to go through lots and lots of school and grant writing in order to get that title. All right. Okay, what if the pathologist is like, I don't have anywhere to raise these flies into an adult, so he just preserves all the maggots. Is the entomologist out of luck as far as ever being able to identify? No. Even if you have maggots, although it costs a little bit more money, excuse me, the maggots can be identified through DNA. Excuse me. So you amplify the DNA, you take the mitochondrial sequence, you throw it into a database online, and the database will spit out, hey, this is a Sarcophagibulata, Formia regina, Chrysomya megacephala, etc., etc. And this is a publicly available database. And this is a really old screenshot. Enjoy going through all of my bookmarks. I mean, look at this. This is great. This is historic. Google Reader, not there anymore. Paul Feinbaum local, gone. Dvorak doesn't have his blog. Reddit, nobody goes there anymore. Dig, oh my goodness. There, and we all had a good time. I know people still go to Reddit, but I found that I just like to look at pictures. So I just go straight to Imager. All right, now that we're done talking about what we do before we fall asleep, and Twitter, the analysis. Okay, so you're the forensic entomologist. you've received all of these. Let's say it's larvae, okay? Because the maggots are probably the most common, if the body's found after 7 days. All right, you find these larvae, you've determined the species, Chrysomya megacephala. Okay, now what do you do? Well, there's a couple different ways to estimate the time of death. Number one, you could look at just the succession of insect fauna. We're going to come back to this later because it's more general and it doesn't matter as much if you can do the second one. The second one is you estimate the age of the immature insects. All right, we said eggs are rarely deposited on the corpse before death. All right, so we want to estimate the post-mortem interval, which is the minimum amount of time. So usually when a post-mortem interval is, why do I keep losing my cursor here? Sorry, for the on-campus classes, I've been doing a lot of complaining out loud this week. I don't know. I think it's just getting towards the end of the semester. Okay, it's the minimum PMI. Okay, I'm going to draw this picture one more time. Okay, remember we have the dead body. Okay. Then we have the flies laying eggs on the body, and then we get to the discovery down here. Okay. All right. When we estimate the age of the insects, okay, if the maggot is determined to be 7 days old, then that means there was 7 days between when the fly laid the egg on the body and the body was discovered. That's 7 days. Okay, the reason why this estimate is the minimum amount of time is because we don't know how much time has passed since the body died and since the flies laid eggs. Okay, this is much more difficult to estimate. Now, the forensic entomologist will estimate that. All right, go ahead and throw guesses into the chat box here. Let me give you a situation. Okay, I take a pig. Okay, let's keep it simple. It's not a human. I take a pig out into a field in South Carolina in July. It's a sunny day. And I shoot the pig. Okay, now I got a dead pig. And I sit down on a rock with my notebook. And I wait, start my stopwatch. How long do you think it's going to take flies to find that dead pig and lay eggs on the body? Go ahead and think about it and take a guess. Is it going to take 2 minutes? Is it going to take 6 hours? Is it going to take a day? Is it going to take a week? Does it depend? Man, you guys are good. All right, Jamie says a couple hours, 30 minutes an hour, 30 minutes. Yeah, and the reason I described it that way is that was an actual study that was done, and they found that under ideal situation, under ideal conditions, it does take about an hour. Okay, not very long at all. Because you may think, well, wait a minute, the fly needs to know that that body's dead, right? It needs to start producing some type of chemical that the fly can sense to know that it's not just some dude sleeping. Okay, but within an hour, the flies will lay eggs. So under ideal conditions, this can be very short up here. But if a body's killed indoors, then it may take the flies a little bit longer there, because if it's at night, flies aren't active at night. It might be in the next morning that the flies find the body. All right, so the forensic entomologist needs to estimate this just based on the conditions. And you may find that forensic entomologists agree on this number as far as how old the maggots are, but there may be some disagreement among different experts as far as how long the body was sitting there before the eggs were laid on it. Okay. Now, I mentioned before that flies will not lay eggs on a body before death. The species that forensic entomologists study won't. There is a species of fly called the screwworm fly. And on campus, I tell this, it's a little bit longer story because some hippie had, this was a while ago, had a screwworm fly lay eggs in their nose. And it turns out that the maggots ate their inside of their face until they died. But the screwworm fly is a fly whose maggots will feed on living tissue. Okay. Now, right now, the screwworm fly is not living in the United States. Okay, it is present in Central America and South America. And the real problem is not that the fly lays eggs in people's noses when they're sleeping. Although if you ever go to South America, I recommend sleeping like this. Okay. But it is a large problem with livestock in cattle. I mean, if you have a screwworm fly lay eggs on a sheep and you get this maggot mass on a living sheep, you can't save it. Okay, so you have to put the sheep down. Same thing for cattle in Texas, who when there were screwworm flies in the United States, they were a big part of figuring out strategies how to drive them out. Okay. All right, so 3-day-old maggot means the body has been dead at least 3 days, plus the amount of time it took the flies to find the body. Okay, flies and beetles undergo complete metamorphosis. So when you recover an insect samples from a body, you may be recovering eggs from the body, larvae from the body, pupa from the body. Now, how can you tell the difference between an adult that is hatched from a pupa and an adult that is just flying around the body? Well, the answer is you can't. But what would you find on the body to indicate that the fly has completed this process? In other words, the egg has become a larva, has become a pupa, and the adult has, because the adult isn't going to hang out on the body, right? It's not going to hatch and then just sit there and go, well, I guess I'll wait here and for a forensic entomologist to collect me so they can determine how old I am. When we came upon the body, we saw a fly sitting behind a miniature birthday cake and looked like it had 2 candles in it. Everybody knows that flies celebrate their birthday every month. So sorry, I'm stalling here in the hopes that somebody will throw a guess. Maybe there's a delay in the recording here. But the answer is it's a hatched pupa. Okay, you don't find an adult, but if you find an empty pupa, then that indicates that it's completed the cycle. Okay, and the body's been there for a while. Okay, so the first step is to identify what stage the insect is in. This is pretty easy. In general, like eggs are around for about 24 to 48 hours, depending on how hot or cold it is. The maggot's around from about 3 days to 10 days. And then from about 10 days to about 2 weeks is the pupa, and usually after 2 or 3 weeks, the pupa hatches into adult flies. Those are just some really rough numbers. I mean, it depends on the species, depends on the temperature. Within the maggot stage, there's a first instar, second instar, and third instar. The maggots tend to go through the first instar and second instar pretty quickly. They stay in the third instar for several days after that. But if you find first instar maggots, then you know that the body has not been there as long as if you find second instar maggots versus if you find third instar maggots. Okay, so this is the first step in, if you do find larvae or these maggots, identifying what instar it's in. And here we see a picture of a fly's or a maggot's butt. Well, I don't know if butt is the right term, but this is the posterior of a maggot. Okay, these are not its eyeballs right here. Okay, these are called spiracles, the breathing apparatus. Dr. Linville, are you telling me that a maggot breathes through its butt? I am. But it doesn't breathe through its butthole. That would be ridiculous. Okay, it breathes through the spiracles, which are located on the posterior, is the correct way of phrasing it. But these spiracles, do you see how there's like 3 footballs here in each, 3 of these brown football-shaped things in each one? Okay, those 3 things indicate that it's third instar. Okay, if it's second instar, then you see 2 of those little footballs. And if it's a first instar, they're just sort of little globules. Okay, so these are what the spiracles look like in the first, second, and third instar. Okay, so that's pretty easy for the forensic entomologist, even for you or I, to pick up a maggot and determine what instar it's in. Okay. All right, so based on the stage, you can take in some general guesses. Okay, like if in here, okay, the egg hatches after 2.8 days, and if we find a pupa, we know it's 22 days old. Eclosion means it's a hatched pupa, then we know it's 37 days old. Okay, just knowing the stage can give us a general estimate. As long as we know the species and as long as we know the temperature. This I'm going to mention a couple times. Okay, What's not shown here in the title is eventually somewhere along here it has the species name of the fly. This table is only good for one species. Okay? So if this is a table for Chrysomya megacephala and you have a Phormia regina fly, you need to go to a different table. Okay? Also notice these numbers are dramatically different based on the temperature. Okay? So the temperature has to be known before you estimate the age of these immature insects. The reason the temperature has to be known is because flies and maggots, they're not cold-blooded because they don't really have blood, but they're not hot-blooded like a human. Okay, like I'm hot-blooded. You can check it and see. I got a fever of 103. I don't know what's happening. Okay, but if you take my temperature right now, indoors, it's going to be what? 97.6. If I go outside where it's really cold and you take my temperature, it's 97.6, right? I'm warm blood. I regulate the inside of my temperature. So all of the metabolic processes that are going on in the inside of my body go at the same rate because they're at the same temperature. But in a maggot, what happens is if you cool down the maggot, the entire body cools down, all of the metabolic processes cool down, and the growth rate cools down. So the growth rate of a maggot is very closely related to the temperature. Okay, we can see here when it's very warm, and this is 35 degrees centigrade. So right here we're at room temperature, 25. Less than a day, those eggs will hatch. Where if it's very cool, 15 degrees, okay, centigrade, which is about, good Lord, don't make me think here, I don't know, in the 40s or something. Okay, 50 degrees, then it takes 3 days. Okay. All right, not only, remember we said that oftentimes it's going to be larva that is found on a body. Okay, the larva, not only do you look at what stage it's in, but you can also look at the length of it. Okay, because at the third instar, it's going to grow a lot. And there's a big difference in the age estimation you can get for like a 7 millimeter third instar larva versus a 15 millimeter third star larva. Okay, so look at this chart. Now this is going to be on your mock case quiz coming up next Monday and on the exam. Okay, you may see a chart and I may ask you a question. Okay. Using this chart, we're going to go through this at the end of the class. You need to know two things. Species, you need to make sure that the species is the correct species. And temperature. You'll notice there's a different line for each temperature. Okay. Now don't get confused. The 15, the little diamond, that's this right here. Okay. How do we know that? Because look, this maggot after 2 days is still very small. And then it's growing 4 days, 6. It's growing very slowly. It takes 18 days to get up to 17 millimeters. Whereas at 35 degrees, the very warm temperature, you can see it hatches and oh my goodness, it grows very quickly. And it only takes about 3.5, 4 days to get up to 17 millimeters. Okay. So just keep that in mind. These charts might not be color-coded. I want you to know the line right here is for the warmer temperature and the line out here is for the colder temperature. Okay. All right, so what's going to affect the growth rate? Temperature, we just got done saying. Hotter it is, the faster these maggots are going to grow. Other factors, if the individual who's passed away does have a lot of drugs in their system, entomologists have found that stimulants, amphetamines, if the maggots feed on them, it'll speed up their growth rate. Depressants, barbiturates, if they feed on those, it will slow down their growth rate. So this can be a challenge for forensic investigators. All right, the day length, all right, it may be a certain temperature, but if you're in Alaska where it's night for 20 hours and daylight for 5 hours, that can affect the rate that a fly grows, versus if it's the same temperature when you're on a much longer day-night cycle. Okay, so That's how you're going to estimate the age with an immature insect sample. Does that make sense? And then the other thing the entomologist wants to check is to make sure that just the types of flies and beetles on the body make sense given their estimate with the immature insect samples. Okay, this corpse is going to provide an attractive environment for different insects, as I've said before, feeding, mating, and depositing eggs. And the corpse is going to an hour after it's dead is going to look a lot different versus 7 days after it's dead, as it starts to go through rigor mortis and bloating and decay. Therefore, flies will be attracted to a fresh body. Okay, different types of insects, beetles, may be attracted to the decaying body. Okay, so just as the body looks different throughout this stage of decay, the types of insects the body attracts is also going to be different. Okay, flies arrive early in the decomposition process. Beetles arrive early or late in the decomposition process. Okay, this is a little chart, succession chart for beetles, and it just sort of shows like different species of beetles when they're present. See, and this is sort of bad. Hopefully it looks better on your screen. But so if you find like sort of, these types of beetles right in here, that may indicate that it's early in the process. Where if you find sort of these types of beetles out here that are found after many, many weeks, then that may indicate that it's much later in the decomposition process. Also, you're looking at the body, the state of decay. But you just want to make sure the species makes sense, okay? You don't want to see a body that looks extremely decayed, but find, 2-day-old flies on it that from a species of fly that usually arrives very early in the decomposition process. It may just set up red flags that maybe something's different here. Maybe the body was frozen and then thawed out or something weird like that. Okay, so the hotter the temperature is, the warmer the body gets, the faster it decays, the faster it'll go through these stages of attracting different flies. So the forensic entomologist has to consider all this. Okay, so that's it for the forensic entomology lecture. We want to conclude with a sample so we can make sure we understand how this process works. Okay, so let's say that we have a dead body discovered at the crime scene at 9 a.m. Okay, so that time is going to be important. If you have a little note card or something, you want to jot this down. Okay, we have maggots found on the body. Some of them, the crime scene unit team is trained on how to collect and preserve these, so they do it correctly. They collect and preserve half the maggots, and they collect some alive and keep them alive in a small cage. Feed them a little bit of water and sugar. That's really all you need to keep the flies alive. Okay, and they are, or excuse me, no, these are maggots. Okay, so you give them a little beef liver. Okay, transfer them to some beef liver and let them grow in the cage. All right. The suspect claims to have seen the victim alive 2 nights earlier. Okay, so what do we want to know here? Okay, if the body has been dead for longer than 2 days, where 2 nights earlier the body was dead, okay, then that means the main suspect is lying. Okay, he couldn't have seen the victim alive. Okay, so if it's a very short time of death, then the main suspect may be telling the truth. But if the body's been dead for a very long time, then we know the main suspect was lying. That's where estimating this time of death can become very important. All right, how are we going to do it? 16 maggots preserved. These are the ones that we're going to measure. Okay, and then we keep some of them alive. All right, let's keep those alive. And what has happened? Oh my goodness, look at this. We've kept them alive, and now they have hatched into this beautiful fly. So this is a blowfly. We don't even need to send this to the genitalia Scandinavian expert. We can look at the wing structure, the bristles, the color, and we know this is very easily. This is Cochleomya macellaria. Okay? We go to the literature. We need to find a chart for Cochleomya macellaria. Okay. Sample investigation. We measure all of these maggots. This is the second piece of information we know. All right. Some forensic entomologists will use the largest maggots, assuming they're the oldest. Some of them will just take an average. There's a debate on which is the best method to use. We're going to go ahead and use the 15 millimeter number. Okay. We want to know how old is a 15 millimeter long Cochleomya macellaria maggot. We have the length, we have the species, we're missing one important piece of information, and that is the temperature. Okay. Now, if this is on, are you listening? Mock case tip time. If this is on a mock case quiz, and I ask you about Cochleomya macellaria, check right here. Make sure it's the correct species. Okay? If I ask you to estimate the time of death for Cochleomya macellaria and I give you a chart for Formia regina, Dr. Linville did not screw up, because one of the answers are going to be, I cannot estimate the time of death with this chart. Okay? And that's going to be the correct answer. So number one, check to make sure it's the right chart for the right species. Okay, now we need to know which line it is. Okay, remember, growing faster should be the warmer line. Growing colder should be the lower temperature line. And we can see here 18 is the lower temperature, 26 is the higher temperature. All right, so we got that going. All we need to know, what temperature was the body found at? 22. Okay, so we're going to use this line. All right. And this is what you're going to have to do. You're going to have to say, okay, 15 millimeters long, that's right here. Go straight across to the red line, as straight as you can, and then go straight down to the best of your ability. You can even get out a ruler and do this on your computer screen. A straight edge or a piece of paper, go straight down. Well, it looks like we're about 95 hours. Okay, what did we say 2 nights earlier, 9 a.m.? So at 9 a.m., the day the body was discovered, okay, 24 is 9 a.m. and 48 is 9 a.m. Okay, well, he said this is night 1 and this is night 2. He said he saw him alive right here. This body's been dead this body's been dead not only 48, 72, 96. He was not alive. Our main suspect is lying. Do you see that? And that's how you solve crimes. All right, so go through this sample investigation. Make sure it makes sense, because questions like this will show up on a quiz or exam. And then that's pretty much it. All right, let me double check to make sure that if any new people have popped in, I'm going to read you guys the attendance. If you have any questions about this lecture or the class, feel free to ask me now. I'm going to leave the lecture running a little bit longer in case somebody does ask a good question that I think will be relevant to other people in the class. And then eventually I'll stop the recording if you guys just want to ask me random questions that aren't as important. All right, Jamie, I got you down. You can go. Jasmine can go. John can go. Ladarius can go. Sarah can go. And Teresa can go. I have you all down and saved. And you should get your 5 points if you don't already have it sometime next week. If not, just pester me about it. All right, so I'm going to stop the recording right now. And if you guys have any other random questions,